Good. That's not too bad. I like it. I like it a lot. Before we move on to generalized chemical reactions, I am going to mention one more specific chemical reaction. What's the one you know already? It's the only way we put things together. It's called dehydration synthesis. Well, it has an opposite. Was that in one of your worksheets? Yeah. What's the opposite of dehydration synthesis? It's called hydrolysis. So let's just mention that. It's the reverse, right? Instead of stealing an H and an OH from two different molecules to join them, what you're doing is basically blasting a water molecule at that big molecule and splitting water in the process to stick H and OH on the two fragments that you cut it into.
Excuse the interruption. Caleb McCool, would you come to the attendance office?
Sorry. It's going to look like nothing was wrong to the remote kids. I had the screen blacked out so they couldn't see anything. So again, instead of pulling water out in the form of H and OH from two separate molecules, you're taking one big molecule that you want to split, throwing water at it fully formed. The water splits into H and OH, displaces the one molecule into two, and sticks the H and OH on the bonds that frees up. We're not going to talk about it a whole lot more than that. We're not going to use it particularly like we use dehydration synthesis, but I want you to know what it is. So any chemical reaction kind of comes down to, are you forming bonds like you are in dehydration synthesis? Or are you breaking bonds like you are in hydrolysis? Forming bonds, breaking bonds. Now, plenty of chemical reactions have both things going on at the same time, right? And so that gets a little more complicated. But ultimately, we can split chemical reactions into two big categories. Do you know what they are? I think you've heard this before. The two big classes, categories of chemical reactions? Yeah? No, chemical, we're talking chemical. Physical changes and chemical changes is what you're thinking of. I know what you're talking about. But chemical reactions themselves. We can say either they're forming bonds or breaking bonds, but because a lot of them have both going on at once, what do we say instead? It has to do with energy. Because some chemical reactions, typically the ones that are breaking more bonds than they're forming, release energy. And some of them, typically the ones that are forming more bonds than they break, are storing energy. Anybody know the words that mean those two things? Energy storing, energy releasing? Kinetic energy is sometimes the type of energy that can be produced. Endothermic is the word we use for absorbing energy when a reaction does that. Endo, of course, means inside. What does thermic mean? Therm? Heat. Good. An endothermic reaction stores energy in its products. And so we can draw a simple graph of the course of an endothermic reaction. And what you're going to find, the way this shows you how much energy is stored, is we're going to start with some reactants. That's what we call the things that go into a reaction. We call them the reactants. And we're going to wind up with some, what's the thing we wind up with? What do you call that? Reactants and products. And we're going to find that the reactants are lower energy than the products are. And the difference between the two is how much energy is going to get stored. There's a little wrinkle in there. reactants, products, before you start drawing, take a look. You notice how it goes higher and then falls down? That's interesting. What that tells you is that to get this chemical reaction to happen, you actually have to put in more energy than the amount of energy that's stored to get it to go to completion. The amount of energy that you need to get the reaction to go is called the activation energy. And that's very important when we start to talk about enzymes. So go ahead and draw that part. And then we'll measure the amount of energy that gets stored, which is the difference between the reactants and the product. In a very real way, you can think of yourself as a big cluster of chemical reactions. You, like you as a functioning living being, are kind of just all the right chemical reactions happening at all the right times, very precisely controlled. They have to happen perfectly, and they do, every single time. That wouldn't happen if it was just this. This is way too much activation energy for you. A piece of bread doesn't just digest itself. Well, I take that back. It does. It just takes a week or two to do it. Right? A piece of bread, if left out in the elements, it will eventually break down. The amylose will eventually break down into glucose in a form you could use it. You don't have 2 weeks to wait. You can't just wait for that thing to naturally break down. You break it down. How do you break it down? You have something that lowers the activation energy, that takes that hill that you got to get. Think of it as almost like pushing a bowling ball over a hill. You got to get it up to the top and over for the reaction to go. You have a special kind of molecule that you make that lowers the top of that hill. You can't lower it below where the products are, but you can make it a lot lower so that reaction happens a lot faster. What is it that your body produces, and you have a specific one for every single different chemical reaction in your body that does that so effectively? Enzyme is the correct word. Enzymes is what you have. Enzymes are the biological version of something you've already learned about. Remember catalysts? Remember what a catalyst is? Something that speeds up a reaction, helps a reaction happen without being consumed in the process. Reusable. Well, you make specially shaped proteins. These are proteins, okay, made of amino acids. Big, long chain, remember? Which of the 20 it is means it folds up into a particular shape. You make certain enzymes. You make certain proteins that function as catalysts. They're called enzymes. And enzymes are just exactly the right shape to, say, grab a starch molecule and stress the bonds just right to split it into glucose molecules. And you have a different enzyme that you make for every chemical reaction that you need to happen at every single time you need it to happen. It's ridiculous. It's so, so complex and cool. Well, here's what it looks like on this graph. With an enzyme, the activation energy is lower. Everything else is the same, right? The products are the same energy. The reactants are the same energy, which means the amount of energy you're storing is the same. But the activation energy is lower. Questions so far? We're going to do the same thing with an exothermic reaction. Can you think of an exothermic reaction? There's one kind of obvious one.
Like the one that gives off like the big like toothpaste explosion, double toothpaste.
I don't know if I know that one. That sounds fun.
I think they put something in like sodium, I guess, and then it just explodes in like these huge colors, and then it like starts giving off like super.
I would say that's what's called a violently exothermic reaction.
Yeah.
The devil's toothpaste. I'll have to look that up. The simplest one I can think of is fire. When something, we call it combustion, right? But yeah, when something catches on fire, that's an exothermic reaction, a fairly violently exothermic reaction. So, but think about this. There's still activation energy. There's still a hill to get over. It just looks a little different than this. It's almost like a reverse of this. Everybody ready to move on? I'll give you another minute or so. All right. So we're going to do the same thing here. You can draw two separate graphs or just do the one like I did with both versions. In an exothermic reaction, look at the hill though before you start writing. Still a hill, right? It's just that now the reactants start higher than the products, and our activation energy without the enzyme is less. It's still there though. Does the wood catch fire on its own? Nope. What does it need? What do you need for the wood to burn? You need some kind of ignition, right? You need a match, you need a lightning strike, you need, I don't know, something, rubbing two sticks together to provide the energy, and all you're doing is pushing it over the top of that hill. All you're doing is adding the activation energy that you need for the reaction to go on its own. Now the difference between reactants and products energy-wise is not the amount of energy stored, it's the amount of energy released. But an enzyme catalyzing this reaction does exactly the same thing. That is, it does not change the reactants, it does not change the products, it changes the size of the hill. Right? It basically lops off the top of the hill. Questions if you've got them. This is pretty abstract though, right? Like you're looking at a graph of energy. It's hard to connect that to the real world. How does an enzyme actually do this is the next question we need to answer. Right? We've said that an enzyme is a protein. Well, proteins are always big long chains of amino acids. You know what those look like. They're the right ones of the 20 amino acids to make a shape that folds up into a shape that does a job. What does the job look like for an enzyme? This is where it gets kind of cool. This is abstract reaction chemistry. Let's look at real 3D objects. Oh, activation energy, lower. I guess put that in. Sorry. Get ahead of myself. So enzymes. Now in the physical world, they are proteins. Understand that enzyme is a type of protein. Not all of them. There's plenty of proteins that do plenty of other jobs. This is one subclass of proteins. But man, they are important. In terms of reaction energy, we can say enzymes lower the activation energy needed to start the reaction. And we can draw that graph. And this is what allows you to be alive. This is what allows all the right chemical reactions to happen at all the right times. But how does it work? Well, an enzyme has a three-dimensional shape because it is a folded up chain of amino acids. Reactions. Sorry, Rx and S is reactions, NRG is energy. That's just me being lazy. An enzyme has a shape. And that shape typically has some sort of little notch that we call the active site. Some kind of little specially shaped nook called the active site. And the active site is the place on that enzyme where the reactant or reactants fit, very much like a lock and key. In fact, this is sometimes called the lock and key model of enzyme function. And by the way, I just threw a new word at you, which is substrate. Substrate is what we call reactants when an enzyme is involved. And so the reactants will have a shape that, I don't know if I can do this with my finger or not, that kind of precisely, it looks like a nose, matches the active site on the enzyme. And when the substrate, which is this, meets the enzyme, which is this, and kind of floats in and locks into that active site and creates what we call the enzyme substrate complex, depending on which kind of reaction it is. If it's a bond breaking reaction, fitting into that active site will just kind of stress the bonds in the substrate just right so that it cracks in half into two pieces. If it's the opposite, if it's a bond-forming reaction, the two pieces will float into the active site, and it'll stress those and smush them together just right so they form bonds. Okay, so they get brought together, these substrates, or split apart by the enzymes at the active site. And there's a little in-motion version of what I just drew, much better than I just drew it. So in terms of reaction energy, you know what that graph looks like. But in the real world, this is what's happening. The thing is floating in to fit inside the thing, which breaks the thing in half. Not as complicated as it sounds, right? Or the thing floats in two pieces, just reverse that, and the thing floats in, two separate pieces float in, and they get smushed together, and they come out as one thing. But that's how enzymes do what they do. That's how they make these chemical reactions happen. Different things can affect enzyme activity. Temperature can really affect all proteins. When they break apart, typically, yes. This is an exothermic reaction. When bonds are broken, it usually means that energy is being released. And when bonds are formed, it typically means that energy is being stored. Yes. It's not foolproof. There can be more complicated reactions where you have to look at the net amount of stored or released. But yeah, that's a good rule of thumb. There's a reason that you are 98.6 degrees. I don't know if it's the reason. Actually, there's some cool research going on about that right now, that being that hot actually kept our ancestors from growing fungus inside them. Fungus don't like heat. But it is true that almost all of your enzymes are built so that they function perfectly at your 98.6 degrees. Much hotter than that, and they're in trouble. Except for one kind. You had a question about this. Some of you answered it perfectly. It was about there's certain enzymes or certain cells in your body that function well at just a couple degrees higher. Yeah? Yeah, it's the enzymes in your immune system. They go to town when they're a little too hot, which means that fever is not something that's wrong with you. Fever is your body turning up the heat on purpose so that your white blood cells can knock the crap out of the bad guys. Bad guys don't work as well. They're attuned to your normal body temperature, right? So kind of a cool response. But temperature, not something we change much. pH also stays very steady. Turns out the way you control your enzymes typically, and this is enough to give you a headache, is with other enzymes. I think that will do for a Tuesday. Geez, it's a lot.